Genetic material means that yes, you guys said that it has some information. Second thing, it can go from one generation to next generation. And third thing, it should be stable. Okay, if not stable, what is going to happen? Mutations. Okay, if genetic material is not stable, then what there is a possibility of what? Mutations. Imagine one day one person is getting born normally, some other day they have wings or something else, right? Some other features, physical features, because of mutations. So it has to be stable. It should not undergo mutation. There is a term we use, retrovirus. What is meaning of retro? अरे retro तो you know ना. हाँ हाँ correct मैम. Old. Retro means old. Or retro songs, retro movies. So retrovirus means old virus, or we can say primitive virus. Primitive viruses, they had genetic material what? RNA. So they don't show central dogma, they show reverse central dogma, right? But RNA was unstable, or it has too much of instability. So it ultimately, slowly with respect to time, it modified into double-stranded DNA. So the first genetic material, if you have to ask, it is what? RNA. Later on, we got DNA. That is a different thing, right? Next thing, the genetic material, it should follow Mendel principles of inheritance. Mendel ke there are certain laws, so it should inherit in a principle of Mendelian way. So first thing we said, it has information, it should be stable, okay, and it should inheritance in form of Mendel way, right? RNA is also known as ribonucleic acid. RNA is also genetic material, but RNA ko, like, we are not considering as a predominant genetic material. Predominant means now in most of the species it is DNA which is the primary source of information. It is not RNA. Why? Because RNA is unstable. So over the period of time, like I said, majority of organisms, they show stability and they change their RNA into DNA, right? But the point is that how we should get to know that DNA is genetic material. So genetic material ka first property we said that it will be getting inherited from parents to offsprings. है ना? So if something is genetic material, the first principle should be it should grow from one generation to next generation. Okay, sorted. So there was our scientist Frederick Griffith. In 1928, he performed one experiment that is called transforming experiment. Okay, so he performed experiment with Streptococcus pneumoniae. Streptococcus pneumoniae causes what? Pneumonia. Okay, there's one more bacteria which can cause pneumonia, that is Haemophilus influenza. But here we are working with Streptococcus. What is meaning of Streptococcus? Oh, Satvik is answering. Yes, Satvik. Huh. Shape of bacteria, fine sir, but which type of shape? Spherical shape. And septo means? Comma is sir, vibrio. Strepto means chain. So spherical bacteria ka long chain is forming what? Streptococcus. Okay. So we don't have to go in that much detail, but we are using Streptococcus pneumoniae, and there are two strains of Streptococcus pneumoniae. We have R strain and we have S strain. Just a minute. Yes. So we have two strains of Streptococcus. We have S strain and we have R strain. R strain, you can see the surface is kind of smooth, and rough strain pe there is no smooth surface. Okay. We believe that R strain, they have a protective covering of mucopolysaccharide. Okay. They have a protective covering of mucopolysaccharide. Because of that, when we eat antibiotics also, or when we take in antibiotics also, they cannot kill our bacteria. If they cannot kill our bacteria, the bacteria will cause disease, right? And the strains of bacteria which causes disease, they are called virulent strains. So we are calling it what? Virulent strain. This bacteria or this strain, R strain, it does not have a protective covering. So when we eat antibiotics, what is going to happen? It will die. and it will not cause any disease. So it will be what? Non-virulent strain. Simple thing. So ignore all the facts. Just keep one thing in mind. R strain is causing what? Disease and is virulent. And S strain. S strain is causing disease and it is virulent. R strain is not causing disease and it is non-virulent. Logical? If I'll be injecting S strain in your body, so what is going to happen? Disease, pneumonia, right? R strain may nothing because antibiotics will kill R strain. Logical? So obviously I cannot do experiment with you people, so I have to take mice. So same thing. Griffith, he did experiment with the mice. Okay, just I'll switch off the sound because I have to ask questions from you also. So, like I said, we have two strains, rough strain. They have protective capsule absent and they are what? Non-virulent. We have smooth strain and they have a protective capsule and they are what? Virulent. So, what we are doing now, where is our mouse? R strain, right? We're injecting into our mice or mouse, whatever you want to say. What will happen as per you? Mice will survive or die? Die. But R strain, na madam, R strain to is what? Non-virulent, na? Right? So mice will survive. Abhi, he will walk also. See? So next we have S strain. Logically, what should happen? Die. The same thing will happen. Life is unpredictable. है ना? One time he survived, now he has to die. Imagine for people like us, mice is dying. Sorted, mice died. Now, what we are doing? This is our S strain. S strain ko we are heating. This is S strain and we are heating it. So now all the proteins which can cause disease, they died. Okay, so this is your heat killed. This is what? Heat killed S strain. All the disease causing proteins we have destroyed. So logically, it should cause disease or not? No, everyone is no, na? So this is heat killed S strain and injected into the mouse, and again our mice survived, which is logical also, right? Okay, this is last condition. What we are doing here is we are mixing our heat killed S strain and we are mixing our R strain. Both they were not causing disease. है Heat kill also not causing disease, R strain also not causing disease. So logically, our mice should survive. So how you can say it died? We'll see first. Proof is important. If you do not see with eye, don't believe it. So this is our mice, and finally this is our fourth condition. We're injecting heat kill plus R strain, and in this condition, our mice, yes, he died after few days. Now point is very simple that in first condition R strain, it was logical mice will not die. S strain mein mice has to die. When we have done the heat killed, mice also did not die. But when we are mixing heat killed and R strain, why our mouse is dying? Logically, तो should not, है ना? Any idea? Anything? Acha, one more diagram I'll show you. When we injected rough strain, okay, and mice, obviously it will live. When we took the sample of blood, na, we found what? Rough strain only. When we injected smooth strain and mice died, so when we took the sample of blood after death of mouse, we found what? S strain from the body. Okay, last, when we're taking heat killed, here also we find what? Heat killed S strain. So here we find what? R strain. Okay, here we found S, here also we found heat killed S. But in case of last condition, when we injected heat killed plus R strain, logically we should find both. But we were able to find what? Live S strain, not R strain. What we are able to find? Live S strain. So we said, not we said, Griffith said that something, okay, something from heat killed S strain, something from heat killed S strain, it entered inside what? It entered inside R strain, and it converted R strain into what? Now that something Griffith didn't knew. What is that something? And that something he called what? Transforming principle. Something was called transforming principle. So if I have to redefine it, so Griffith mentioned transforming principle from heat killed S strain. Okay, it entered inside R strain and converted it into live S strain. And now we have live S strain, so S strain can cause disease. Sorted? This is the basic thing. But the important thing is that we have to find out that what is this something, right? So imagine you have a test tube. Okay, just a minute. Or yes, so we have this test tube. Hana, in this test tube, we have only what? I should start from here, or okay, fine, no issues. Let's start from here only. So you have this test tube. We have removed all the things, lipids and sugars. So what we are left with? We are left with DNA, we are left with RNA, and we are left with what? Proteins. Fine. And this test tube, it contains what? Heat killed S strain. Heat killed S strain. Now this culture, whatever it is in the test tube, we divide it into three parts. Okay, this is first test tube. We can say condition 1 or case 1. Now in this test tube, I added enzyme, let's say protease. So protease will destroy what? Protein. So we will have what? We will have only DNA and we will have only RNA. Okay, so I used enzyme, which one? Protease, to destroy proteins. In case 2, imagine I'm using DNase. Okay, so here we had what? We only have RNA and we have protein. And last condition, that is our case 3. Here I'm using RNase. Okay, so obviously this test tube will have DNA and this will have what? Protein. Fine. And in this conical flask, here we have a conical flask. Okay. In this conical flask, let's assume we have R strain. Okay. Here also we have R strain, R strain, and here also we have R strain. Sorted? So you have taken a test tube, and in that test tube we have heat killed bacteria, heat killed S strain. We have removed all the things, lipids and sugar. We are left with DNA, RNA, and proteins. We divided content of this that test tube in three parts. In one test tube we added protease to destroy proteins. In one we added DNase to destroy DNA, and in last one we added RNase to destroy RNA. Now we'll be adding, or we'll be mixing the content of what these test tube into this conical flask. So in case 1, I added all the content of this into this conical flask. What happened? After some time, okay, this R strains, they modified into what? S strain. After some time, what happened? R strain modified into S strain. This gives kind of idea that protein is not a genetic material, or it was sorry, transforming principle was not protein, right? If protein was transforming principle, so this would not have happened. So either it is DNA or RNA. When what we did, when we added content of this test tube into the R strain, now this does not have RNA. What happened? Again, R strain changed into what? S strain. Now in first case, it was proved protein is not genetic material. In this case, it is proved RNA is not genetic material. So maybe it is DNA, right? Final confirmation we got from case 2 when the content of case 2 was added into what our R strain conical flask, there was no change because it contains what? RNA and proteins. So somehow we got the idea that there might be possibility that transferring principle is what? DNA. Got the experiment? Got the idea? But people were still not able to believe this thing, but fine, I'll come to that part later on. Same thing is mentioned here. So we had 3 scientists. Name of scientist is super important. The question is also very important. They used to ask us multiple times that who did the biochemical characterization. Biochemical characterization means who identified that our transforming principle is DNA. So it was Avery, MacLeod and McCarthy. These 3 scientists. Okay, they did biochemical characterization of transforming principle and they said it is what? DNA. Prior to this experiment, sorry, prior to this experiment, it is mentioned that our protein was considered as genetic material. Okay, so by this experiment, we got a little bit of idea that might be possible DNA can be genetic material, but still people did not believe it. So there was requirement of one more experiment, which was done by Hershey and Chase. 2 scientists were there, Hershey and Chase. They worked with E. coli and bacteriophage. Okay, how I will tell you, but I believe this thing is sorted. R strain may no disease, S strain may disease, right? Heat killed may also we have no disease, and when we are mixing, we are getting what? Disease. Why we are getting? Because some part of S strain is entering into R strain, which is modifying them into S strain, and that some part is called transforming principle. Later on, 3 scientists said this transforming principle can be DNA.